Hello students and my dear friends, myself Madhu Vanja. In this session, we'll discuss some of the important bacteriology multiple choice questions. First question, false statement regarding meningococci is. So what is meningococci? Neisseria meningitidis. Option A, meningococci are gram-negative cocci and they are arranged in a pairs and they contain the adjacent slides. And second option, fresh isolates of meningococci are capsulated. Third option, meningococci are motile. Fourth one, they were first isolated by Welsh album from the spinal fluids of the patient. So here the right answer is option 3, meningococci are motile. So that is a false. So remember one point, all cocci are non-motile, like which are gram-positive cocci and gram-negative cocci. So cocci, they doesn't possess the flagella, so that's what they are non-motile. So meningococci is non-motile, it is not motile. Next question, which among the following are incorrect regarding the pneumococcus? Option A, they occur in pairs. Option B, they are gram-negative. Option C, they are capsulated. Option D, they are readily stained with the aniline dyes. So here the right answer is they are gram-negative. They are that is incorrect. Actually, they are gram-positive cocci. They are gram-positive cocci. So here the right answer is they are gram-negative cocci. The incorrect. That is an incorrect statement. Next question. Streptococcus pneumoniae is the causative agent of option A, pneumonia. Option B, otitis media. Option C, meningitis. Option D, all of the above. Here the right answer is option D, all of the above. So here Streptococcus pneumoniae, they causes pneumonia, which is lung infection, and they causes otitis media. What is mean by otitis media? That is the infection of the middle ear, and they causes meningitis. Meningitis means inflammation of the meninges. What is meninges? Meninges is the outer covering layer of the brain. Next question, on gram staining, pneumococci can be observed as option A, gram-positive lanceolate diplococcus. Option B, gram-positive cocci, they are arranged in the chains. Option C, gram-positive cocci occur in the clusters. Option D, gram-negative cocci arranged in the pairs. Here the right answer is option A, gram-positive lanceolate diplococcus. So after gram staining, pneumococci appears as gram-positive lanceolate diplococcus. What is meant by lanceolate diplococcus? Lanceolate diplococcus means one end of the cocci or one edge of the cocci is round or broad, and another end of the cocci is narrow or sharp. So they are arranged as lanceolate diplococcus. Two cocci are arranged as a pair. They appears purple or violet in the color. Next question, gonococci can be differentiated from meningococci by following sugar fermentation test. Option A, glucose. Option B, sucrose. Option C, mannitol. Option D, maltose. Here the right answer is maltose. So here by using of maltose sugar fermentation test, we can differentiate gonococci as well as the meningococci. Next question, all of the following are causative agents of NGU except option A, Chlamydia trachomatis. Option B, Mycoplasma hominis. Option C, Candida albicans. Option D, meningococci. So what is mean by NGU? NGU means non-gonococcal urethritis. NGU means non-gonococcal urethritis. So here the right answer is meningococci. So meningococci doesn't cause urethritis. It causes meningitis. But if you see other options such as Chlamydia trachomatis, Mycoplasma, Candida albicans, they causes urethritis, non-gonococcal urethritis. Next question, the bacterial sore throat is caused by option A, Staphylococcus aureus, option B, Haemophilus species, option C, Streptococcus pyogenes, option D, Mycoplasma pneumoniae. Here the right answer is option C, Streptococcus pyogenes. So Streptococcus pyogenes causes sore throat. Next question. Crystal violet blood agar is a selective medium for option A, beta hemolytic streptococci. Option A, beta hemolytic streptococci. Option B, Staphylococcus aureus. Option C, Corynebacterium diphtheriae. Option D, Mycobacterium tuberculosis. Here the right answer is option A, beta hemolytic streptococci. So, like a beta hemolytic streptococci, they grow in the crystal violet blood agar media. So generally, streptococci is resistant to the crystal violet. So in the preparation of selective medium, while preparing the blood agar, we add crystal violet. So this crystal violet suppress the growth of other bacteria and they promote the growth of the streptococci. Next question. Puerperal fever is a streptococcal infection of option A, kidney, option B, skin, option C, uterus, option D, heart. So here that is uterus. The right answer is uterus. So puerperal fever, so that is an infection of the uterus. So generally it is an infection after the delivery in the mothers. Next question, identify the bacteria which are oxidase negative and catalase positive. So which bacteria is oxidase negative and catalase positive organism? Option A, Staphylococcus. Option B, Streptococcus. Option C, Neisseria. Option D, Pseudomonas. Here the right answer is Staphylococcus. So Staphylococcus, they are oxidase negative organisms and catalase positive organisms. Next question, drug of choice for MRSA. So what is meant by MRSA? Methicillin resistant Staphylococcus aureus. In case of MRSA infection, which drug is used? Option A, penicillin G. Option B, ceftriaxone. Option C, vancomycin. Option D, cefzolin. Here the right answer is option C, vancomycin. So vancomycin is the drug of choice for MRSA. So in case of methicillin resistant Staphylococcus aureus, we use vancomycin as a drug of choice. Next question, toxic shock syndrome is caused by option A, staphylococci, option B, E. coli, option C, pneumococci, option D, meningococci. Here the right answer is staphylococci. So staphylococci, it produce toxin called toxic shock syndrome toxin. So this toxic shock syndrome toxin causes toxic shock syndrome. Here if you see streptococci, like Streptococcus pyogenes also causes toxic shock syndrome. But here in this option, streptococci is not there. So here staphylococci is the answer. Even staphylococci and streptococci both causes toxic shock syndrome. Next question, interferon gamma release assay IGRA has the advantage over tuberculin test. Option A, positive indicates active infection. Option B has less false negative results. Option C has no false positive results. Option D, it can be used as marker of prevalence. Here the right answer is it has no false positive results. So if we do the like a tuberculin skin test, so in some cases we may get the false positive results also. But in case of interferon gamma release assay, so we do not have any like false positive results. Next question, CBNAT. The CBNAT cartridge-based nucleic acid amplification test. All are true except option A, works on real-time PCR principle. Option B, detects MTB, Mycobacterium tuberculosis complex, DNA. Option C, detects rifampicin resistance. Option D, detects isoniazid resistance. So here the right answer is detects isoniazid resistance. So here first three statements are correct only. They works based upon the real time on like a real time PCR principle, that is RT-PCR principle. It will detect Mycobacterium tuberculosis complex DNA. It detects rifampicin resistance, but it doesn't detect isoniazid resistance. Next question. Which of the following properties are the characteristics of tetanospasmin? So what is tetanospasmin? Tetanospasmin, it is a powerful neurotoxin which is produced by Clostridium tetani. Option A, it is a heat labile protein. Option B, neurotoxin. Option C, it can be toxide. Option D, all of the above. Here, the right answer is it is a neurotoxin. So it is a neurotoxin. It acts upon the nervous system. Next question: which of the following bacteria is known as Hansen's bacilli? Option A, tubercle bacilli. Option B, Bacillus anthracis. Option C, Leprobacillus. Option D, none of the above. Here, the right answer is option C, that is Leprobacillus. Next question, malignant pustule is a term used for option A, an infected malignant melanoma. Option B, a carbuncle. Option C, rapidly spreading rodent ulcer. Option D, anthrax of the skin. Here the right answer is option D, anthrax of the skin. So malignant pustule, it is a anthrax of the skin. So in the malignant pustule, we can see the central necrotic lesion. It is surrounded by the several satellite lesions, which are having the characteristic black smear. So it is called as a malignant pustule. Next question, which of the following bacteria lacks the cell wall? Option A, Helicobacter. Option B, Bordetella. Option C, Mycoplasma. Option D, all of the above. Here the right answer is option C, Mycoplasma. So Mycoplasma, they do not possess the cell wall. It is a cell wall deficient bacteria. So as it is the cell wall deficient bacteria, there is no proper shape and no proper size. So it exists the pleomorphism. Next question, the pathogenic treponema is cultivated in option A, culture media. Option B, viral cell lines. Option D, rabbit testis. Option D, anaerobic culture methods. Here the right answer is rabbit testis. So treponema pallidum, it can be grown only in the rabbit testis. So serial testicular passages, they cannot be grown in any artificial culture media. They can grow only in the rabbit testis. Next question, the type of mycoplasma that can cause genital tract infection. Option A, Mycoplasma hominis. Option B, Mycoplasma genitalium. Option C, Uriplasma urealyticum. Option D, both A and C. Here the right answer is both A and C. They causes urinary tract infection. Next question, Reganlu medium is used for isolation of option A, Bordetella pertussis. Option B, Vibrio cholera. Option D, that is Brucea malae. Option C, Shigella sonnei. That is option A. The right answer is option A, Bordetella pertussis. Next question, in satellitism, the organism that is streaked across the blood agar plate perpendicular to the Haemophilus influenza streak is option A, Anthrax bacillus. Option B, Staphylococcus aureus. Option C, Pneumococcus. Option D, Corynebacterium diphtheria. Here the right answer is Staphylococcus aureus. So here when we grow the Haemophilus influenza bacteria in the blood agar media, actually Haemophilus influenza bacteria, they require the 10th factor as well as the 5th factor which are present in the blood. So but 5th factor is not freely available. To make it available freely in the center of the culture blood agar media, we inoculate the Staphylococcus aureus. And after that, we inoculate the Haemophilus influenza. So this is Staphylococcus aureus, it makes to release the 5th factor really freely into the culture medium. So that 5th factor will be utilized by the Haemophilus influenza. So that mechanism is called as satellitism. So which streak is used? Staphylococcus aureus is used. Next question, the organism that most commonly causes whooping cough is. So what is the causative agent of whooping cough? Option A, Bordetella parapertussis. Option B, Bordetella bronchoseptica. Option C, Bordetella pertussis. Option D, Bordetella avium. Here the right answer is option C, Bordetella pertussis. So Bordetella pertussis causes whooping cough. So that is like a majority of the cases are caused by Bordetella pertussis. And Bordetella parapertussis causes that also causes whooping cough, but that is a milder form. So here the right answer is Bordetella pertussis. Next question, the infections that can be caused by Pseudomonas aeruginosa. Option A, urinary tract infections. Option B, wound and bone infections. Option C, respiratory tract infection. Option D, all of the above. Here the right answer is all of the above. So Pseudomonas aeruginosa causes urinary tract infection, wound, bone infections, and respiratory tract infections. Next question, the color responsible for bluish green due to the Pseudomonas aeruginosa. So which factor is responsible for bluish green color? Option A, pyoviridin. Option B, pyorubin. Option C, pyocyanin. Option D, pyomelanin. Here the right answer is pyocyanin. So pyocyanin is responsible for bluish green hue of the Pseudomonas aeruginosa. Next question, the type of motility shown by Vibrio cholera. Option A, gliding motility. Option B, swarming motility. Option D, darting motility. Option D, flagellar motility. Here, the right answer is darting motility. So Vibrio cholera, it has like a darting motility or characteristic vibratory motility. The name Vibrio comes from vibratory motility. So Vibrio cholera, it has single polar flagella. Next question, the color responsible for bluish, sorry, this question is repeated. That is option is pyocyanin. Next question, concentration of agar used for inhibition of swarming growth. Option A, 2% agar. Option B, 3%. Option C, 6%. Option D, 10% agar. Here the right answer is 6% agar. So generally, when we grow the like a Proteus bacteria, so Proteus is like a highly motile bacteria. So Proteus bacteria is highly motile bacteria. So to control this swarming growth or to control this motility, so instead of 2% agar, while we prepare the culture media, we increase the agar concentration. That we increase the agar concentration to the 6%, so that the culture media will become more solid. Automatically, it inhibit the like a motility of the like a bacteria, or it inhibit the swarming growth of the Proteus species. Next question, microorganism which can cause plague. Option A, Listeria. Option B, Yersinia. Option D, Bordetella. Option C, Actinomycetes. Here the right answer is option B, Yersinia. Yersinia pestis causes plague disease. It is a zoonotic infection. It is transmitted from animals to humans, which means it is transmitted from rats to humans. Next question, enrichment media used for Vibrio cholera. Option A, alkaline peptone water. Option B, selenite broth. Option C, tetrathionate broth. Option D, soya bean casein digestive media. Here the right answer is alkaline peptone water. So enrichment media is the liquid media which has added inhibitory substances. They suppress the growth of unwanted bacteria and they promote the growth of wanted bacteria. So alkaline peptone water, it suppress the growth of other bacteria. It promote the growth of Vibrio cholera. Next question, Klebsiella pneumoniae, they are also called as option A, Klebsiella granulomatis. Option B, Friedlander's bacillus. Option C, Bacillus mucosa capsulatum. Option D, both B and C. Here the right answer is option D, both B and C. So Klebsiella pneumoniae, it is also called as Friedlander's bacillus and it is also called as Bacillus mucosa capsulatum. Next question, the selective media used for Salmonella is option A, cetrimide agar. Option B, Wilson and Blair media, bismuth sulfate medium. Option C, tellurate blood agar. Option D, brain heart infusion agar. Here the right answer is Wilson Blair bismuth sulfate medium. So this is a selective media. So selective media is a solid media which has added inhibitory substances. So they also suppress the growth of unwanted bacteria and they promote the growth of wanted bacteria. Next question, serological test used for detection of enteric fever. So what is enteric fever? Simultaneous infection of typhoid and paratyphoid. Option A, VDRL test. Option B, Weil-Felix test. Option C, Paul-Bunnell test. Option D, Weidel test. Here the right answer is Weidel test. So Weidel test, it is a serological test which is used to detect the typhoid and paratyphoid fever. Next question, Salmonella are option A, gram-negative cocci. Option B, gram-positive bacilli. Option C, gram-negative bacilli. Option D, gram-positive cocci. Here the right answer is Salmonella. They are gram-negative bacilli. Next question, rapid laboratory diagnosis of dysentery uses. Option A, immunofluorescence test. Option B, precipitation test. Option C, agglutination test. Option D, complement fixation test. Here the right answer is immunofluorescence test. So dysentery, it is caused by Shigella dysentery. Bacillary dysentery is caused by Shigella dysentery. So here Shigella dysentery, it is easily identified by the immunofluorescence testing. Next question, prophylaxis of shigellosis. So what is shigellosis? It is a whole spectrum of disease caused by Shigella dysentery. Option A, vaccination. Option B, passive immunization. Option C, sanitary measures. Option D, all of the above. Here the right answer is sanitary measures. So there is no vaccination. So vaccine is not available for Shigella and there is no passive immunization also. So here the right answer is sanitary measure. So here by following the hygienic conditions, we can prevent the Shigellosis. Next question, the vaccine used for enteric fever is option A, BCG, option B, HPV, option C, TAB vaccine, that is TAB vaccine, option D, DPT vaccine. So here the right answer is option C, TAB vaccine. If you see BCG vaccine, it is for prevention of tuberculosis. HPV, human papilloma virus, it gives protection from cervical cancer. That vaccine gives protection from cervical cancer. And DPT, diphtheria, pertussis, tetanus. It is a triple vaccine or combined vaccine. It gives protection from 3 diseases. But if you see TAB vaccine gives protection from typhoid fever and paratyphoid fever. Next question, the causative agent of bacillary dysentery. Option A, Salmonella. Option B, E. coli. Option C, Shigella. Option D, Staphylococcus aureus. So here the right answer is Shigella. Here Shigella dysentery causes dysentery. Remember, bacillary dysentery is caused by Shigella dysentery. Amoebic dysentery is caused by Entamoeba histolytica. So amoebic dysentery is parasitic infection. Bacillary dysentery is bacterial infection. Next question, E. coli and other species of bacteria which found in the intestine help our body too. So they are commensals actually. Option A, break down fats. Option B, they make vitamin K and vitamin B complex. Option C, they absorb oxygen. Option D, remove the waste material. Here the right answer is, so these E. coli and other enterobacteria species, they produce vitamin K as well as the B complex, which will be helpful for our body. Next question, E. coli was discovered by scientists. Option A, Thomas Escherichs Weimer. Option B, William Escherichs. Option C, Theodor Escher. Option D, Bruno Latour. Here the right answer is Theodor Escher. Thank you.